Thank you. What I've got here is some liquid nitrogen. As you know now, it's at minus 200 degrees centigrade. And I've got some frozen mouse embryos in here. And we're going to see how they might be fertilized and how one might make, as it were, a test tube mouse. Because in this lecture, which is about the right stuff, what I want to think about, or what I want to talk about, is how we can make different cell types. For example, if Frankenstein's going to make his angel, he might have to make the halo, and for example, he's got to find a suitable cell to make the halo with. So what I want to look at is where the different cells in your body come from. Now, they come from the fertilized egg, and so what I first of all want to do is to see how one might fertilize an egg. Could I have a volunteer, please? I feel very mean about this. Could you come, please? Thank you. And I'll tell you what I want you to do. If you come and stand over here. What's your name? Naomi. Naomi, what I've got in here is some frozen mouse embryos, and I'm going to take the top off. Have a look inside. It's okay. Have a look and see. Because down there at the bottom, what you've got to do is take out those frozen embryos. And the only danger is if you drop these in, there's a danger that the thermos flask will break. So I think, have a feel about that. They're not the most elegant of instruments. Okay, do you think you can get that out and get it into here? Let's see if you can. You can look what you're doing. Don't rush it. You got it? Terrific. And into there. Terrific. That's absolutely fine. Well done. Hold on, don't go away, because you're going to fertilize too. Hold on. So let's put those there. So those are the frozen mouse embryos. And so when one talks about test tube babies and in vitro fertilization, that's exactly what we're going to do now. Now, these take a little while to thaw because they're actually extremely cold. And Amata has already prepared, under the microscope, some that we've thawed out a little earlier. Can we just have a look at those? You just wait here for a moment, because what we're going to do is we'll fertilize them in a moment. Let's see if we can find them. I'll go over here. There we are. There we are. There is a mouse egg that's been taken from the frozen canister, warmed up, and it's surrounded by other cells that really look after it. Now, what we're going to do is I'm not, Naomi's going to, add some sperm. And now it's quite hard to see fertilization under the microscope. And part of the reason is the eggs are very big and the sperm are very small, okay? But nevertheless, so what we'll do is we've looked at that. After you've added the sperm, we'll concentrate on the sperm, because if we try to see both, you're simply not going to see it. So we've got the eggs there, and here we've got some sperm. And Amata, if you bring that over here, and Naomi, you come on me this side here, and Amata will show you how to fertilize them. Do you think you've got enough there?
Yes.
Okay. Well, you've actually fertilized now. Well, the sperm have now got to swim in. Thank you very much indeed, Naomi. Thank you very much indeed. We're going to... And we're now going to have, only one of the millions of sperm are going to get in, but that's very hard to catch. Oh, there's a beautiful one swimming there. Now, there are absolutely millions in there, and of course they've got these large heads. And of course, that contains the genetic information. Thanks very much, and thanks very much, Naomi. That's nice fertilization. And just to show you what it looks like in man, could we just have the next? We've got a sequence here, which is slightly speeded up, of showing what happens in the human. Here you can see very nice sperm again, speeded up. Lots of them attach, only one gets in. And there are special mechanisms to prevent that. The egg, for some reason, becomes rotated by the sperm. Fertilization has now occurred, and development begins. Now, what you've seen there, or what one finds with fertilization, the important thing is not getting development to start, because really you can get the egg to start quite easily. Any good old kick, any old nasty treatment will almost get development to start. The importance of the sperm is it's the male's genetic contribution, because in the head of that sperm is the genetic information that is required to join up with the genetic information in the egg, which is in the nucleus. And the important thing to realize is that it is in fact the genetic information, the genes, the chromosomes, in the nucleus of the sperm and in the nucleus of the egg that really determine what you're like. Whether you're male or female, whether you've got brown hair, yellow hair, long noses, short fingers, all your characteristics, almost all your characteristics, are determined by those genes in the nuclei. And particularly, as we'll see, whether you're male or female. Now, if you actually have a look at what a cell looks like, let me remind you what a nucleus looks like. Because if you look at these moving cells, the nucleus is this clear area here with the black spots in. Here's a nice cell, and there's the nucleus with some material inside, probably chromosomes. Here's a nucleus here. There's another nucleus there. In other words, the nucleus is a bag inside the cell and separated from the rest of the cell, which contains the cytoplasm. And the genes are in the nucleus. That's the crucial thing to remember. And what happens at fertilization is that sperm brings in fact, there's nothing much else to sperm other than the genetic information in the nucleus in the sperm, and that comes into the egg. I have here a model of the cell. It's a rather mushy model of the cell, as you're going to see. And there we can see the nucleus that I was referring to there. If we just go back here, there's a nucleus, there's a nucleus, there's a nucleus. And here's my nucleus here. I've got it there. And this is the cytoplasm, and this is the cell membrane here. And what I want to do is to take out that nucleus, and I need another volunteer. If you'll come round here. What's your name? Mark. Mark, I want you to sort of make a vigorous cut. If you take the scissors, don't be frightened, nothing. I'll take this off. What I want you to do is put in here and zip it up there, and then cut it up here, and then I want you to take out, it's a bit messy, no matter. That's what cells are like. So be vigorous. Great, yes, that's what I like to see. And the other side, nearly my fingers went there, that's okay. Terrific. Okay, think you can get out the nucleus now? Terrific, lovely. Hold on, very nice. Hold on, I'd like you to clean it up a bit, please. I'll move this away, because we're going to open it. Oop, nearly gone. Shall we try and, shall we both try and open this here? Let me put on my glasses so I can see what I'm doing, because I know that there's a little, I can see it. You've got to. And what I want you to do, would you take out from there what we've got? Hold on. Do you think you could lay out the pieces just gently on there? And what those are, of course, are the chromosomes. Normally, you can't see them very clearly in the cell, but when the cell divides, they condense into these rod-like objects. And you can see them. You don't need to make them too neat, you just spread them out. And normally, we've got 46 in our body. Every cell in our body has 46 chromosomes, 23 from your mother and 23 from your father. Well, thank you very much for opening up the cell for me and showing me that there are chromosomes inside. Just let me show you another picture of the chromosomes. And this is what the chromosomes look like when the cell is dividing. They're condensed, 23 from your mother, 23 from your father. And you can pair them up, and that's shown here. Here they're paired up. And you see, everything pairs up very nicely except for two. This one here, which is known as the X chromosome, and this little one down here, which is known as the Y chromosome. It's these two chromosomes that determine whether you're male or female. All eggs have an X chromosome. Every egg has an X chromosome. The sperm that we saw brings in either an X or this rather teensy Y. If it brings in the little Y, then you're going to be like me, male. If it brings in an X, you're going to be a female. So this is from a male. Could I have the next one where you'll see where it didn't bring in a Y, but it brought in another X. So all females have two X's. And that's the difference between male and female, just that small little difference in genetic information. So we have the fertilized egg. We are now going to have to look at how we get all the different cell types. And I'm going to use as a model for the nucleus in all these cell types this rather splendid jukebox here. And I need another volunteer. No, I'm going to come from this side this time. Because you've got about 250 cell types in your body. You've got skin cells. Now, I want you to come and stand over here, and I'll tell you what we're going to do in a moment. And if you could just make sure that you stand back so as with other people, because they need to see the jukebox. This is a mechanical jukebox, not in one of your modern ones where you don't see the mechanical parts. And what this jukebox has got, it's got records in here. And I'll tell you why it's got records and what those records represent. The records in here represent proteins. Proteins are the really important parts of your body. If you met a cell on a dark night and you wanted to know what sort of cell it was, I'll tell you the question you ought to ask it. You ought to say, what sort of luxury proteins do you have? And if it's a sensible cell, it would turn around and say, well, my luxury proteins are hemoglobin. And then you would know it was a red blood cell, because hemoglobin is the protein that carries the oxygen in the blood and is in the red cell. If, on the other hand, the cell said, my luxury protein is keratin, then you'd know it was a skin cell, because the luxury proteins of skin cells is keratin. So all the instructions for these proteins, all the instructions for these luxury proteins, and I'll tell you in a moment why I call them luxury proteins to distinguish them from household proteins, all the instructions to make these are contained within the nucleus. And so this is the nucleus of a cell. And what we have here, in the form of the records in here, the instructions to make all the proteins in the body. However, these skin cells don't make hemoglobin, and my blood cells don't make keratin. But just let's see what happens. Let's actually try an experiment. What we'd quite like to do, what I'd quite like it to make in the first instance, is something that's made in the pancreas, and we'll see what happens if we try for insulin. Insulin is something that's made in the pancreas. Can you see which one you've got to press?
B7.
If you do that, let's see what happens. And then stand back. Oh, we better reset it. Let's try it again. Okay, if you stand back and let's see what happens. So we've given the cell a particular signal, and we'll see if the instructions for insulin are here.
Insulin, insulin, insulin.
So it's got a very, oh, I'm glad you like the tune, Top of the Pops there. So if you give the right instructions to the nucleus, you'll get out the instructions for insulin. Okay. But there are lots of other proteins. And I'm also told you that there are all sorts of household proteins. And the household ones are really rather boring proteins. They're the ones that every cell has. And so they're the ones to keep the cell dividing, to keep the cells moving, to keep the cells alive. And they're common to large numbers of different cells. So we're not terribly interested in. Let's have a listen to the ones that are characteristic of the skin, keratin. What number is that one?
B3.
Could you do that, please? Oh, you're sharper at these games than I am, I can see that. And so, in your skin cells, this is the song that the nuclei is. Rather brief, but to the point, I say. So as I tell you, there are about 250 cell types in your body: lens cells, skin cells, pancreas cells, pituitary cells, all of them with their own special luxury molecules. And which one is made is determined by which record the nucleus plays. and we'll play just one more. And that's haemoglobin that carries the oxygen in the blood. Reset. What's haemoglobin?
B5.
Terrific. And here we go for the top of the pops. No haemoglobin and you're really in trouble. I think that's rather a fancy tune. Which one did you like most? Insulin. Thank you very much for that. That's very kind of you. So what you've got to think of is the nucleus of the cell containing the tunes, the instructions for all the proteins in the body, but only some of them are played. And of course, the proteins aren't made in the nucleus, but they're actually made in the cytoplasm, and that gooey stuff that we played with. Okay. Now, the last protein that we played there was haemoglobin. And haemoglobin is an extremely important protein. It's made by red blood cells, and it's the one that carries the oxygen in the blood. Everything's fine so long as the record is a perfect one. As long as the instructions are absolutely spot on, there's no problem. But if there's even a small error, a mutation in that genetic information and those instructions, things can go wrong. And there's a whole variety of diseases that arise, for example, that the record, the instructions for haemoglobin are just slightly wrong. There are diseases like thalassemia, but a much more common one is sickle cell anemia. And there's just the tiniest change in the tune. You'd barely, if you listen to that rather nice little jingle of haemoglobin, you'd hardly hear it. But it does make an enormous difference to the protein. And instead of the protein folding up in the correct way, it folds up inaccurately, forms rods, and it deforms the cell, and that has rather serious effects. Because here, instead of having nice, smooth, normal red cells here that flow through the capillary, delivering oxygen the way they should, the cells change shape and they sickle because the protein is folded up wrongly. And when they get to very narrow capillaries, the blood doesn't flow properly because these sickle cells begin to clog up the capillaries, and so flow, look, there it's blocked. And that can be extremely painful, and it also means that you're not delivering enough oxygen to the tissues. And so you go from a faulty record on the one hand, through the protein, through a change of shape in the cell, through a change in circulation, to actually a disease known as sickle cell anemia. What we want to know then, and what you want to know, what Frankenstein wants to know, what everybody really wants to know, is what determines which of the tunes are going to be played. And I'm going to use two cells to illustrate this the first time. What I've got here in this bottle are cancer cells. Don't worry, they're quite safe. They're not going to leap out and do anything with you. I claim that I could drink these cancer cells and still be quite okay. I'm not going to try it, but I think in principle it's okay. These cancer cells grow in suspension. They're being stirred. This orange is merely an indicator to show that the acidity, that the pH is correct. Millions and millions of them in here. And these cells, or their great-great-grandparents, have been growing for something like 25 years, and they're called HeLa cells. And they're cancer cells, and they're called HeLa cells because they were taken from a tumor, from a cancerous tissue of a woman called Helen Lane about 25 years ago in order to study cancer cells, and people have found them very convenient cells to study, and they've been growing ever since. So these are very active, in fact, if anything, too active cells. And I'm going to represent that and its nucleus by this nice shiny 50p piece in this hand here. So that's my HeLa cell, okay? Then there's another cell, a much duller cell compared to that, and that's the chicken's red blood cell. Now, the chicken's red blood cell is truly dull because unlike the nuclei of these cells here, they're doing nothing. No household tunes, no luxury tunes, silence. And here is the nucleus, my little 1p piece, my nucleus of the chick red blood cell, doing absolutely nothing. No luxury tunes, no household tunes, no nothing. And compared to the vibrancy of the HeLa cell nucleus. Now, what you can do now in cell biology is to put these two nuclei into the same cytoplasm. And you can do this by bringing the cells together. So here are my cells. I'll bring them together, and I can fuse the membranes between the two cells so that the two nuclei end up in the same cytoplasm. The way one does that is that one can use chemicals, or one can use a particular virus known as Sendai, and that will make the cells fuse. So what you've got now is the two nuclei in the same cytoplasm. And that's shown over here. There's a HeLa cell, and here's a HeLa cell that's fused with a chick red blood cell. And there's the sort of rather dull chick red blood cell nucleus, and here's the rather large, very active HeLa cell nucleus. And the result of that experiment is that, in fact, this nucleus, which was not doing anything before, when it's in this cytoplasm, becomes reactivated. It doesn't make haemoglobin, but what it does, it begins to make all the household proteins and all the household tunes associated with chickens, which means that the cytoplasm of these cancer cells can get household tunes playing in the nucleus, in the jukebox nucleus of the chick red blood cell, which up to now has been virtually, well, not virtually, completely inactive. And what that shows, of course, is that the cytoplasm on which the nucleus rests is crucial in determining which button is pressed. So now we can do a somewhat more dramatic experiment. And let me tell you what that experiment is. Here I have another mushy model of a cell. But this one slightly different to the other one, because this one I want you to think of as having been taken from the intestine, from the gut of a tadpole. So one's taken a tadpole, taken out the gut, separated the cells, and here it is, and there's its nucleus. Okay? And what I want to do is so this is, of course, singing the tunes or making the tunes appropriate to gut cells, making the luxury molecules, the luxury proteins that gut needs. And over here, I have a fertilized egg, a frog egg just about to set off into development. But its nucleus sits right at the top here, and I can effectively enucleate that egg with a little ultraviolet irradiation. I can shine an ultraviolet light here, and I can kill that nucleus. So let's pretend I've done it. I've killed this nucleus, and so here I've got an egg with no nucleus. Okay, so now I can do the experiment. I can go along, and I can zip this open with great, very mushy. Take out the nucleus here. Take out the nucleus of the intestinal cell. Excuse me when I just clean it up. There it is. And what I can do is that this nucleus, which was singing gut cell tunes, I can take it along, and I can put this nucleus into this enucleated egg. And the question is, what's going to happen? Is this egg going to turn into a gut cell? Or, because the cytoplasm is the thing that's determining what the nucleus does, will it say, stop singing gut tunes, play egg tunes to me? In other words, will this now support development? And let's have a look actually what happens when one does that, which one does, in fact, with axolotls. Here are some axolotls that come from America, in particular Mexico. They're really larval amphibians. And there are its eggs. And if one takes an embryo, one's separating out now some of the cells in order to get their nuclei. There are the cells, and one's going to take their nuclei, and then one will take the eggs, do that ultraviolet irradiation, so they're effectively enucleated, and then one's going to inject into them the nucleus from one of those cells. And there's the injection. And then one can have a look at what happens. And what happens is that development, instead of it turning into a gut cell or something like that, it actually develops moderately normally. In fact, really quite normally. There it's going through a cleavage stage. then it's going to form a blastula, and it's going to actually fold its neural tube in a moment. In other words, that nucleus is now responding to egg cell cytoplasm and is behaving as if it were a normal egg cell nucleus. So all these have identical genetic information, and those are identical quintuplets. Or no, sextuplets. I can't count how many. Yes, there are 6 of them. They're all identical because they've all got exactly the same nuclei inside of them. And that's cloning. And that's very important. That's very important for 2 reasons. It shows 2 things. First of all, it shows that this cell from the gut here, the nucleus from that gut cell, has still got all the records inside. It's still got all the genetic information. If it didn't, you wouldn't be able to get those larvae. You wouldn't be able to get adults. So everything is still intact in that nucleus. Secondly, it shows you that the cytoplasm of the egg can make that nucleus behave in an appropriate way. And thirdly, it shows how you can get a large number of identical animals by cloning. So that, for example, in principle, you may not wish to, and Frankenstein, I doubt whether he would, you could make, in principle, a large number of copies of me or you, because you could take my gut cells, take out the nuclei, find someone to donate eggs, enucleate the eggs, put the nuclei in, get someone to agree to carry them through to birth, and you could get a whole lot of identical, well, not me, but perhaps you. I was never such a good-looking baby, I have to say. But you could imagine you could get a large number of people like that. And I don't know whether you read in the press or you've seen the movies, there are claims that multimillionaires have made copies of themselves, that people are raising armies of people, of identical men in order to invade the world and so forth. And the question is that really possible? Well, in principle it is probably. In principle it probably is. But in practice, it's extremely unlikely. And let me tell you why it's extremely unlikely. That while it works quite nicely with amphibia, when you come to something even like the mouse, it hardly works at all. In fact, it doesn't work at all. And just let me show you what the experiment is with the mouse. And here's the mouse egg, and you are now on a very small scale. This is about a fifth of a human hair. These are very fine instruments, and you can see the mouse egg here being held and a single nucleus with a bit of cytoplasm around it being injected. And so that's how one does the experiment with a mouse egg. Now you will say, can we go along to the mouse and take a gut cell, take out its nucleus, inject it into the mouse like that, and get a mouse? The answer is no, you can't. And in fact, even if you go to the 4-cell stage, remember the mouse egg divides once and then twice, if you take a nucleus from very early on in development and inject it, development is aborted. It just doesn't work. So if that doesn't happen at that very early stage, the possibility of taking cells from much later stages, like my skin cells or something like that, are extremely remote, and it's just not going to work. And in any case, who wants to make a whole lot of me? Or maybe you think you would like to make a whole lot of you. Just remember, I think this is extremely unlikely. I think this is just one of those fantasies that journalists like to spread around to make us frightened. Makes a good story, but as far as science is concerned, extremely unlikely. I certainly would be anxious about cloning, and I wouldn't like to pursue that, particularly in man. However, you probably realize that there are clones amongst us, that it's already happened. And of course, what we have are 2 people here with identical nuclei in them, and that's Lisa and Vicky. And if you'd come down, Lisa and Vicky are twins. And of course, they're from a single fertilized egg, and therefore they have identical genetic information. I always forget which is Lisa and which is Vicky.
Okay,
Lisa. Now, they are identical twins, but yet they clearly are slightly different. I think, now that I've met you before, that I can recognize the difference between you. And I think that's very important, because what it means is, although they've got identical jukeboxes inside every one of their cells, they haven't ended up looking exactly the same. And that means that there are other factors, there are random elements, developmental noise, if you wish, which actually change development even though the genetic instructions are identical. Now, you may say that this has only occurred since they were born, that one eats more or less or does more sport or something like that. So what we thought we'd look at is something that wouldn't be affected, as it were, after they were born, and that's their fingerprints. Because, in other words, if one committed a crime, would your fingerprints be exactly the same? And we'll find out now. Come, let's do your fingerprints, and we'll find out. I'm not the greatest fingerprinter in the world, I have to tell you. We're going to take your forefinger, and let me do that there, and we're going to roll. Yes, if you press, I can see that you're terrific. And which are you?
Okay.
I can't read without my glasses, so you do it for me. That's really not bad. Thanks very much. Lisa, will you do it? Should we do the next one? Terrific. As close as we can to the other. Thanks very much. Ted, you need a cloth. Hold on. Here's some stuff to clean yourself with. It's rather mucky. Let me put this under the microscope, and I'll focus it, and we'll see how similar your ones are. I just want to have a look. Oh, yes. Do you want to come and have a look at your two fingerprints? They're a bit like mirror images, actually. Have a look down the microscope, and we'll have a look on the BP here. Do you think they look the same?
No,
They're like, a bit like mirror images, but they're not identical, are they?
No.
Well, that's a relief in a way, I suppose. If you have a look at them here, they're slightly like mirror images, aren't they?
Yeah.
But not identical at all. Well, that shows that there are at least some other factors, and are probably true in the brain too, that not everything is genetically determined. Thank you very much indeed. Thank you. We can now look at another way of making animals, or look at a way of making animals with different genetic constitutions. And the way that we can actually do this is to have a look at how we might make mice with 4 parents. Normally, as you realize, you have 2 parents. These mice have a great propensity to jump out of here, and I'm going to just be a little careful. These are perfectly normal, respectable mice, 2 parents each, mother and a father, but we've got black mice and we've got white mice. But what we can do is we can make a mouse or mice, oh, it hasn't got a no, in which each offspring has 4 parents. And the way we do it is we take the embryo of a white mouse, take the embryo of a black mouse, and fuse them together. There's the black mouse embryo, and that's the white mouse embryo. Pushed together and fused with fine needles, and actually, when put back into a mother mouse, that will develop into a very nice mouse. And the question is, what sort of a mouse is it going to develop into? Let me ask you. So I fused a black mouse with a white mouse, and we have here the result, which I shall reveal to you in a moment. Do you think half are going to be black and half are going to be white? What do you feel? You must have views. Guess.
Grey. Black and white. White spotted black.
White spotted black.
Grey.
Grey. You don't think some of them are going to be white and some of them are going to be black?
Black and white.
Striped. What about looking like a Dalmatian? No. Well, let's actually have a look. Here's the result. And since I'm just frightened it's going to run away, and there it is. And as you can see, hold on, I'm going to actually pick it up. And there it is. It's really a striped mouse. It's not really a grey mouse. It's got patches. And of course, what's happened, it's got 4 parents, this mouse. I don't know whether you regard that as a good or a bad thing. For most of us, 2 is more than enough, as they say. Okay, I'm going to put you back on there. And I think if I don't put this on, don't want to catch its tail, it's going to run away. Did you see that clearly enough? And so what you get from that, experiment is that you find that there is a mouse with 4 parents, and its cells really express the genetic constitution of both, of all 4 parents, really, and so that you get a striped mouse. Now, I can give you, oh, they're getting, excuse me. It is about development after all. Here's another example of a chimera. Just let me explain to you what I mean. Those there were a chimera. I think you should remove these, Bryson, to the privacy of the cage. Thank you very much indeed. They've certainly put on a performance for us. By a chimera, I mean an animal that is made up of cells of different genetic constitution. And historically, the chimera was an animal with a lion's head, a goat's body, and a serpent's tail. And that striped mice, or one can make striped mice like that, which are chimera. And here's another rather strange beast. This one here is a chimera, and it's looking rather strangely at us. And you can see it's got a goat's-like head, but it's woolly, like a sheep in certain parts. And in fact, this is a sheep-goat chimera, and we would have brought it to you here in person, but he's just too old to travel, and so it wasn't really able. So he's aging a bit. He's not as sprightly as he is there, and therefore we weren't able to bring the animal to you. But you can see the sort of sheep-like characteristics, and if you look at the head and the horns, if we can see that, they are goat-like too. And so there you've got a chimera. So one can make sheep-goat chimeras. It's not as frisky anymore. Now, I think that sort of does generate anxiety in quite a lot of people, because I think the idea of making a chimera, for example, between, let us say, a human and a chimpanzee, I think that is something that we all find somewhat distasteful, and I certainly find it distasteful. And I don't think anybody wants to do that sort of experiment. And my guess is that once you go across species, although sheep-goat works, I don't think it's actually going to work. Now, even though you may think that it is something that's unattractive, I have to tell you that we have chimeras amongst us. That is, there are those amongst you who really are made up of 2 quite distinct cell types. Every female here in this audience and throughout the world is a chimera, not with a lion's head and a goat's body. But if you remember, if we go back to what made the difference between male and female, you will remember that females have 2 X chromosomes. We poor males only have 1 X chromosome. And you, all the females, have 1 too many. And therefore, 1 of every X chromosome has to be inactivated. And when the embryo, when all the females were about 100 or 1,000 cells in their embryo, randomly there was inactivation of 1 or other of these X's. So every female throughout the world and here is made up, half their body have, or half their cells, not half their body, half their cells have 1 X turned off, and on the other half, the other cell is turned off. And let's have a look at what the implication of that can be. Because imagine that on these X chromosomes, there were genes for making orange pigment and black pigment, yes? Then if and let this one be the orange and this one be the black. If the orange one is on, then it's going to look orange. If the orange one is off and the black one is on, it's going to make black. And you can see, I think you can begin to see, well, what sort of effect that's going to have. And what we've got is a group of people here with cards, orange and black, and if you could hold this up, let's see what sort of a pattern we get if we randomly inactivate the X chromosome in these cards. So we've got orange and black, and what I want to see what happens is that we randomly turn them up. Could you hold them short side up? Short side up, yes. That's it. So, so they're oblongs like that. Okay. Now, could you lift them up? Can you rotate them? Try rotating. That's terrific. Yes, just keep turning. That's it. Just keep turning round. Well, very good exercise, and I like it. That's very nice indeed. Okay, stop. And could you hold your cards in front of you? So what we've done is we've randomly inactivated half of the chromosomes. And you see about half of them are black, and well, slightly more than half are black, and the other half are orange. And that's the sort of pattern that we'd expect to get if you randomly inactivated the X chromosomes in an animal whose X chromosome, one X could make orange and the other one could make black. Now, I wouldn't have told you that if there wasn't an animal which actually does that without twiddling cards and does it very naturally. And that, of course, is Tabitha. It is Tabitha, yes, which Gareth is holding here. Can we see? Oh, look at Tabitha's beautiful body. And what has it got? It's got patches of orange and patches of black. And Tabitha is a tortoiseshell cat. And the reason why Tabitha has these patches of black and patches of orange is exactly the same reason that we saw here, and that is that the cells have been, the X chromosome has become randomly inactivated, and that's why you get the pattern in that way. And she is a female, isn't she? Yes. Good. Thank you very much indeed. And don't ask me why she's got some white markings as well. That's another story altogether. Thanks very much. Now, while you're sitting here, while you're actually here, you may think that development is over. Yes, we know you're growing. But that's not true, even if you've stopped growing. Development is occurring right as you sit there now. Just think about your skin and think about your blood. You know that when you get sunburnt, the skin peels off. If you burn the top of your mouth, you lose the skin there, and that's replaced. Where do you think it's being replaced from? Your skin is growing all the time. And let's have a look at this here. The skin is a very thin layer. The epidermis, anyhow, is a very thin layer, just covering the whole of your body. And if you magnified it, you'd find that there was a group of cells at the bottom, that's at the bottom of the skin here, which were growing. Cells, when I say growing, I mean dividing. They are multiplying. They leave this layer and begin to mature, like going to school perhaps, or leaving nursery school, begin to change, to wrinkle a little bit, begin to make keratin, one of their luxury proteins, flatten, and these are the cells that are on the surface of your skin. So if we just go through that again, multiplying cells, maturing cells, cells flattening, making keratin, and these are the cells here that are peeling off the end of your skin. Okay. And then there's another example here. Let me show you that, is that if you think about your blood cells, while you're sitting here, you are making blood all the time. And there are a group of cells, stem cells, which all the time are dividing and maturing and giving rise to all sorts of different cells. They give rise to lymphocytes, they give rise to red blood cells, they give rise to macrophages, other white cells, to cells that help with the clotting of the blood, and there are all sorts of complicated cells in blood. And they all come from the single dividing stem cell. These cells mature in different ways. Now, it's by knowing about this that you can begin to understand what radiation damage does. Do you remember the story with Chernobyl? And we have here one of the victims from the Chernobyl, and as you probably know, when you're exposed to radiation, then you actually lose your hair and you get rather nasty radiation burns. Now, the nature of the burn that you get when you suffer radiation damage isn't the burn of the kind that you get from a kettle. The reason why you get a burn is that radiation stops cells multiplying. So what happens is the cells keep on leaving your skin, but there are no cells to replace them because the cells aren't multiplying anymore. And so you get ulcerations and you get what effectively looks like a burn. What's more, if you think about your bone marrow, the stem cells no longer are multiplying. What the result is, none of these cells are being made anymore. And when one talks about bone marrow transplant, what one's talking about replacing the stem cell so is that this whole production can occur again. So as you're sitting there now, your cells in your epidermis and your skin are making cells that are coming off, and your stem cells are dividing, and all these cells are dividing to give rise to these differentiated cell types, the different cells of the blood. It's understanding this that also gives you an insight into cancer. And here I have a mouse that has spontaneously developed a tumor. Let me take it. I don't know if you can see it. It has a large tumor at the back there. Can you see that large lump on the screen? Can you see this large lump here? That is a tumor that it spontaneously developed. There it is there. It bit me during rehearsal, so I'm keeping my fingers away from it. Now, what has happened, what happens in cancer, is that a particular group of cells continue to divide in an uncontrolled way, and that can give rise to a tumor. But you can also get cancer of the blood, and that's known as leukemia. And let me try and explain that to you, that there you've got a solid tumor here, but you can also get a tumor of the blood known as leukemia. Let's have a look, not at these cells here, but I want to look at one particular group of cells. You can have a cell derived from this stem cell passing through a series of phases in which the surfaces are different and which the cells are maturing to become the mature lymphocyte. It's rather like, well, you might think this is the cell at nursery school, then it's at doing its CSC, then it does its O levels, then it does its A levels, then it just gets tired and turns into a lymphocyte over here. And the lymphocytes on the whole are not dividing. Okay, and that's a nice story, a nice picture of maturation. What happens with cancer of the blood is that something goes wrong and the cells are at one cell, just one cell. All cancers practically start with one cell going wrong. So of all the millions of cells in the blood, just one goes wrong and it gets stuck here. And instead of going on maturing through CSC, O level, A level, it gets stuck. It remains immature and it goes on multiplying. So it doesn't develop into a nice, respectable, mature lymphocyte. And the blood becomes stuck with cells at this stage of development. And you can see this in normal blood. Now there's normal blood, and there's a nice mature lymphocyte. Gone through school, nice life history, nice and stable. However, in someone with leukemia, there are many, many immature lymphocytes in here. And you can see the blood full of them, and you can see them in high power. Here they are. These are exactly the cancer cells filling the blood with these immature cells, just like I've shown here. And if only one could find some way of changing these cells so as they will go through the normal maturation pathway, one could. It's an unlikely way of doing it, but it's a possible way. If only one could correct these defects, they would no longer be cancerous cells, but they'd end up as normal lymphocytes, and that would be an important way in which to get things to happen. Now, it is possible to persuade cancer cells to behave normally. And there's one particularly dramatic example. There are very few examples of this, but there's one particularly dramatic example, and that is there are a group of cells, of cancer cells, that come from the germ cells, which, if they get into a mouse, they'll cause a tumor. However, you can do something else with them. Here are the cells. These are the cancer cells which will normally form a tumor if they're in a mouse. But if you inject them into the early embryo, here's the early mouse embryo being held, and here are the cells, not nuclei, cells being injected into the mouse embryo. So we're now making a 4-parent mouse again. They don't give rise to tumors at all, but in fact simply take part in the normal body building of the mouse, and so that you end up with a mouse not unlike the one that we were speaking about a moment ago. We end up with a 4-parent mouse, a black and white mouse again. And so that when you take these cancer cells and put them in the right environment where they can be controlled, that is in the early embryo, they behave like perfectly normal cells. And I think that's rather a dramatic example of control of how cells are actually going to behave. Now, it's one of the triumphs of genetics that one can now begin to investigate at the molecular level how the jukebox is effectively controlled. I'm going to talk now about 2 organs. Just let me remind you, every cell, every tissue in the body has the same nucleus. And one of our problems is what turns on which bit in which cell. Why do you make, for example, a protein called elastase in the pancreas and growth hormone in the pituitary? Let me just show you where the pancreas is. I've got to take this apart, probably just dissect it out. Pancreas is behind here. Let me take out the heart. Okay. Take out the liver. Take out the stomach. Oops. And there's the pancreas. Pancreas secretes all sorts of important things into your stomach. And one of the things it secretes is a luxury protein called elastase. Okay? And that breaks down elastic tissue. And then I want to talk about another region, the pituitary, which is in the middle of your head. Take out the brain. And right in there, in the middle of your head, is the pituitary gland. And that makes a luxury protein called growth hormone. And that determines how big you grow. And it's one of the techniques of genetic engineering that you can introduce more growth hormone into different cells nowadays, with the result as follows. That you can actually inject DNA, genetic material, into mice, so that instead of making the normal amount of growth hormone, you can make more growth hormone and get this very large super mouse here. And that's simply due to the injection into the egg of a large or of a small quantity of additional growth hormone, which is then turned on in all cells. And look at this very large super mouse here. And now I'm going to try and illustrate that to you more clearly with a sort of game. And what we're going to look at is these 2 regions, the pancreas and the pituitary. And if I could have the genes, please. And here we've got the elastase gene. Would you stand there? And here we've got the growth hormone gene, if you stand here. Now, normally, the product of the growth hormone gene is growth hormone, and that should be in that cracker there, we hope. And the product of the elastase gene is in this cracker here. But these genes, I regret to tell you, can do very little on their own. They just sit there. Because what they need in order to pull the cracker to make their product is a gene that normally sits just next to them. And could I please have the promoters? And here we have the elastase promoter. If you come and stand here. And here we've got the growth hormone promoter. Okay. Now, between them, they could now actually get the right tune and make the right product. Okay? Not yet. Not yet. I should remind you that if we're thinking of this, here we've got the pancreas, here we've got the pituitary. There are all the other genes in both of them, but we're only going to concentrate on these here. Okay. Now, in order for the growth hormone gene to work, and in order for the growth hormone promoter, for them to get together and do their act, as it were, they need a signal. And could we have the signaler, please? And there's the pituitary signaler. Would you stay there, please? And here's the pancreas signaler. Would you come around here? Okay. Don't do anything yet. Hold it. Now, this is the pituitary signaler. And when the growth hormone promoter hears the pituitary signal, they can pull the cracker. Just let's try the signal first. Don't do anything yet. Terrific. Absolutely terrific. And the same here, we've got the pancreas signaler, which the elastase promoter hears, and then it activates the elastase gene. Just let's hear it. Terrific. Okay. And now we better find out what actually happens when that works. I want to tell you, you are only interested in that signal. Never respond to anything else. And please never respond to any other signal. Okay. Let's see what happens. Pituitary signaler, let's have it. Keep going. Aha! And what do we find here? Without much, we find growth hormone. Okay, thank you very much. No, don't go away. That worked rather nicely. There's growth hormone, and that was the result of the pituitary signal. Could we try the elastase signal, please? Or at least, I don't mean the elastase signal, I mean the pancreas signal. Try once more. Aha! Aha! And what we've got here is elastase. Okay? Well, that's nice normal development, and that's very, well, that's fine. Now, okay, that doesn't tell you very much. Now, it's one of the real marvels of recombinant DNA, of the modern molecular biology, that you can introduce genes to other genes who have never, ever met before. And we're going to do that now. So I'm going to take the growth hormone gene, and I'm going to, would you just go around there? And if you could, no, you stay there. If you could just stand back a little bit, back there. Terrific. Okay. And now if you just stand there, and I'm going to take the elastase promoter. Could you go around there, please? And your name is?
Sarah.
And your?
Colin.
So we've got Sarah. Have you ever met before?
No.
Terrific. That's what I expect. And that's the same with the genes. What you can do with genetic engineering, you can bring together genes that have never been together before. So now you're together, and you've got your cracker. Would you like to come around this side, please? Okay, no, that's it. Hold on, let's get rid of all this. Remember what you respond to. Let's have the pituitary signal. No. Do you respond to the whistle?
No, I respond to the horn.
Then don't do anything, please. Nothing happens. After I told you not to, I'm not surprised. Okay, let's go along to the pancreas side. And let's see what happens now. Let's see what we've got inside. You're the gene, you get it out. And what we have here, of course, we've got the growth hormone gene, we've got the elastase promoter, and we're making growth hormone in the pancreas, which has never been made there before. And it's for reasons like that that one ends up with, and that's why one can get supermouse like that. Okay. Now that we've got supermouse, tomorrow what we're going to have to look at is superfly. Thank you very much indeed. Well, thank you very much indeed.